I'm Jed Singer, a scientist in Gabriel Kreiman's lab. We're trying to understand how the cascade of processing throughout the visual part of the brain leads to our perceptions. Visual information progresses very quickly through the brain, but traces of it linger for a relatively long time. As we move through a dynamic world, might those traces allow us to mentally combine or keep track of the parts of an object seen at different times? We're recording activity in the brains of people while they identify pictures that have been broken into pieces and presented asynchronously. This helps us to understand when and where in the brain things viewed at different moments come together to provide a coherent picture of the world around us. This understanding will then guide the creation of biologically inspired computer programs that can mimic our flexible powers of sight. While performing these experiments, we used a computerized camera system to track what the subject is looking at. Here, you can see a small green circle jump to each new point that is fixated while watching Home Alone 2. We work with epilepsy patients who have, for clinical purposes, electrodes implanted in their brains. In addition to providing crucial clinical information, these electrodes allow us to monitor human brains while subjects perform various tasks. Here, we show the readouts from 3 electrodes placed over the temporal lobe while the subject watches a clip from a cartoon.